Material.
Hello, I am Jacob Underwood, and I'm going to be introducing the answers for number 1. So, which end is the 5' end? Which one's the 3' end of the first one? And we're looking at the left side being 3', the right side being 5'. What is the sequence of the DNA template DNA strand? And that would be the same one that you receive on the top. That is the template. And the sequence of the coding DNA strand would be 5' to 3', because you're doing it the inverse when you're taking them, because U to A or A to U, and then G to C. And it would be AC, GAA, CGG, TTT, AGT, AGGG, CT, ATCT. And then we were supposed to draw an image and be sure to label the mRNA, the template DNA strand, and the coding DNA strand. We have our image right here. We have our template strand being like right here, our coding strand and our mRNA strand, and we have like recopying that. And then the mRNA will be going out into the cytoplasm, hopefully into the ribosome to have like, but it might get the wonderful journey of DNA. And then for number 2, TFIID binds directly to the TATA box, which, while sigma factor binds directly to the -10 and -35 sequences. Guanylyl transferase is a eukaryotic enzyme that links the 5' carbon of one nucleotide to the 5' carbon of another nucleotide. Group 1 introns use the nucleoside guanosine as a cofactor during catalysis, while group 2 introns use adenine as a cofactor during catalysis. CPSF is an endonuclease that assists in terminating transcription in eukaryotes. In lesson 9, we learned about the general transcription factors, GTFs. TFIIB is a GTF, which acts as a bridge subunit to link TFIID to TFIIF RNA polymerase 2. TFIIH is a GTF, which acts to phosphorylate and activate RNA polymerase 2. The spliceosome is composed of subunits called SNRMPs. These spliceosome units are composed of 2 components, proteins and SNRNAs. Thank you very much. Answer up.
Right. So I am just going to do number 3 really quick here. So this question concerns proteins involved in transcription and RNA modification. So the first one is for 2 proteins that are in either prokaryotes or eukaryotes that act as helicase, and so are specifically during transcription. And it's TFIIH and RNA polymerase, and then indicate 1 protein in prokaryotes that acts as helicase to separate RNA from DNA during transcription, and that's the Rho protein. And then 3 molecules in eukaryotes that function during RNA modification and ribozymes are group 1 and group 2 introns and the SNRMPs. And the 1 protein factor in prokaryotes that is a transcription factor is the sigma factor, and the 1 enzyme in eukaryotes that synthesizes long strings of identical RNA but does not use as a template DNA for RNA synthesis, and that is the poly-A polymerase. That's it for me.
Yeah, thanks, Nathan. All right, I'm Josh Allred, and I'm doing the 4 and 5. So in question number 4, we have a eukaryotic gene, and it's called my favorite gene. What do they call it? So,
What gene?
your favorite gene.
Oh, okay. I guess it has to be your favorite.
That's what it says. I'm just reading the script.
Suck up.
So it has 4 introns, and it wants us to draw the pre-mRNA that's hybridized to be the DNA copy. So we have that up here. We have 1 R loop, and then with the mature mRNA, we have 4 intron DNA loops, and then R loops in between there. You can see that there. And then it wants us to draw one with mRNA that's hybridized to DNA. However, the 5' most intron has a defective disorder, so that would be over on this side. So there's no loop there, so we only have 3 loops. And then D, we only have 2 loops because we have 2 defective introns there. All right, and then the introns get degraded, and then the exons are transported to the cytoplasm. So now we have question number 5. Which of the above is found in Bacterium E. coli? We have group 1 intron, -35 sequence, and that's in the DNA. And then we have NUS-A and RUT, and all those are only in prokaryotes, so that's how you know they're only in E. coli. And which of them are found in yeast? That's a eukaryote, so we'd have SNRPs, the GC box, the -25 sequence, enhancers, silencers, which either amplify how fast it's getting replicated, or silencers which slow it down, and the TF2D, which helps initiate it. And then which are involved in transcription initiation, like I said, TF2D, -25 and -35. Which of the above is involved in transcription termination, NUS-A and GC box, and then which of the above is a DNA sequence. So that would be the -25, -35 GC box, and group 1 and group 2 introns. Thank you, and that's all we have for you today.